Thank you very much. So now we have a natural progression in how the morning is going with Nicholas Schork from the Venture Institute. We'll be building on this to talk about causal designs for efficacy. And I guess my question for you is, what type of evidence is needed?
Well, we'll find out.
We'll find out.
Yeah. All right, well, it's a real pleasure to be here, and I thank the organizers for inviting me. I have a couple of topics. Sorry about that. How do I go back? How do I go back? I must hit the wrong one. Oh, there you go. And then the pointer is this.
The big green, yeah. And that's
Got it. Sorry about that. 4 topics I want to talk about. First, just raise the issue of having a need to reconsider clinical trials designs in the modern era with all the emerging technologies that researchers have access to. Talk about the N of 1 and aggregated N of 1 trials and their motivation and how one designs such trials. Talk about personal versus population thresholds, an emerging concept given the ability to monitor people over time. And then talk about the idea of actually vetting algorithms for matching nutritional interventions to individuals. A couple of disclosures. I do consult for some pharma, as well as being the founder of a few little companies. And then at about 15 years ago, I was actually a member of the Food Nutrition Board. So the need to reconsider clinical trial designs in the modern era. We all know that there are some pretty severe and dramatic changes that have occurred at the FDA in the last little while. Most of those have been based on new legislation. For example, the 21st Century Cures Act introduced by Obama at the tail end of his tenure in the White House. In that legislation, what is actually called for is a reconsideration of the RCT, and maybe the substitution of RCTs with data summaries and real-world evidence. This is going to have pretty big implications for how one brings evidence to the FDA as part of a filing. They put out a number of reports kind of advocating these changes right here. One thing I'm not going to talk about, but it might be of interest to the community, and I've talked about this in other venues, is there's actually a movement afoot to do away with phase 3 and phase 4 clinical trials and substitute them with what people are referring to as a learning system, which is just a way of collecting data on people, say through electronic medical records, and then mining that data to look for trends that might be clinically useful for patients going forward. This was advocated in a recent article by at least a number of people, but one of the co-authors, Sean Causal, is actually associated with the FDA, and I'll mention him a little later in my talk. So what are N of 1 trials? I've had the opportunity to comment on these in the press recently. Also had the pleasure of working with the FDA to talk about them about 5 or 6 years ago. I will mention that their focus is really on trying to get a patient better rather than vetting an intervention. There's a huge difference. You could imagine having multiple interventions, not knowing which one might be best for your patient. And so you could use crossover designs, as I'll talk about, to test each one as long as the condition is not acute and life-threatening and you have the luxury of testing them out. You can see which one performs optimally for the patient. And that way, not hedge your bets with any one, but just let the data do the talking. I will mention, too, that N of 1 trials have been used for all sorts of purposes, including nefarious purposes. You probably know a little bit about Cambridge Analytica and Facebook, where content was given to individual users to find out which content appealed to them most. This had a pretty dramatic effect on the election, or so it has been argued. I'm going to talk about using them in the health context, obviously. So how do they work? Well, they're pretty simple. Let's say you have some measure that you want to optimize for a patient, and there's some equipoise going on. You don't know the optimal treatment. And let's say your output here is weight, blood pressure, something else. What you do is take a baseline, and then you provide an intervention to the patient and record their clinical measure. Maybe wash them out and give them an alternative intervention. Do that some number of times, recording the clinical measure throughout the cycle of alternating treatments, and then read off, to the degree that's possible with data, the treatment that worked best. And you could imagine having multiple patients subjected to such a trial where one may respond to one intervention and another may respond to a different intervention. Also, you could imagine collecting lots of data on these individuals to make claims about correlations between the changes in the clinical parameter and, say, biomarkers of interest. This could be fairly sophisticated depending on the nature of the measures you're using. You can have positively correlated phenotypes, negatively correlated phenotypes, uncorrelated phenotypes, et cetera, et cetera. These trials have been pursued in the literature. You'll be able to read about a few of them. I tabulate a couple here. I will say that many of these legacy N of 1 trials were not done with a great deal of rigor, so you've got to take them with something of a grain of salt. There was guides put out recently on how to conduct N of 1 studies, focusing on, among other things, what are called single-patient open trials, which don't require an IRB. This is just if a physician is in, again, a state of equipoise, they can practice the art of medicine by doing whatever they think is in the best interest of the patient, and that might mean alternating therapies to see which one works best. Actually, this idea is not entirely new. Hogben and Sim in 1953 proposed this, so I encourage you to go look at this kind of precedent-setting article that they had put out. You could leverage all sorts of emerging wireless devices to look at clinically meaningful parameters on patients subjected to an N of 1 trial. This is literally about leveraging big data for individual patients to make claims about their health status in response to interventions of all sorts. I do want to emphasize that the power of these N of 1 studies is not due to the number of subjects. It's due to the number of observations that you make and the study designs that you're using to really make causal claims about the influence of an intervention on a clinically meaningful parameter. So fundamentally different than the way historical population-based trials have been pursued. Had a recent paper discussing different N of 1 trials designs. I'm not going to go through these in any great detail. Time won't permit. But I will draw your attention to one design having to do with the sequential nature. So let's say you want to minimize the amount of time that a patient is on an inferior therapy. As you're building up evidence, say in favor of one therapy versus another, you could sway the balance of time that the patient's on what looks like the more beneficial therapy, and in that way engage in a more ethical trial in the sense that you're minimizing the amount of time someone's on what is looking like an inferior therapy. These trial designs can be optimized. Again, I won't go through this in the interest of time, but just say my colleagues and I, as have others, have tried to optimize these designs to, again, minimize the amount of time someone is subjected to such a trial, but still be able to collect enough data to make objective claims about the efficacy of a particular intervention. In one context, you might have a finite number of measures to make on your patient, and you want to optimize the distribution of the collection of those measurements. So if you had two interventions that you were contrasting here, you might put the patient on one intervention and then put them on another. It might be more optimal to actually kind of rotate the number of times they're on each intervention, in this way optimize the amount of data that you'll generate that isn't influenced by the overt serial correlation between the observations. If you're collecting data on a single individual, there'll be strong correlations between the measures. And so what my colleagues and I have shown is that you can actually, with an assumption about the strength of the serial correlation, optimize these designs to maximize the power that you'd have to actually bring out one effect, say, over in another. You can also aggregate N of 1 studies. So let's say you have 10 patients that you've subjected to an N of 1. You could look at the trends that they exhibit over the trial and group them into clusters and then ask questions about what those individuals might have in common. Maybe they share a genotype, maybe they share an environmental exposure. And in that way, you might have some way of determining the best intervention for future patients without having to subject them to an N of 1 trial. Many N of 1 trials have been pursued that are of relevance to the nutritional field. I'll just draw your attention to a few pursued by Victor Herbert, whose name came up yesterday. He actually subjected himself to an N of 1 trial and then had another person that he studied who had been subjected to severe dietary constraints due to economic reasons, and then gave him folate supplementation to kind of determine the benefits of folate and the appropriate dosing. More recently, there was an N of 1 study by Golding here, who actually tried to match what Herbert did and subjected himself to severe dietary restrictions and then gave himself folate supplementation to see if he could kind of come up with the same conclusions that Herbert had did. So it was a remarkable study that he had pursued. I want to dive into another topic here, this personal versus population thresholds. It's a pretty simple concept, but I think it's one that will resonate with many people in the nutritional field. So let's say you have a particular biomarker. I'm going to use cholesterol because it's just an easy one to walk you through. And let's say I had 25 individuals whose cholesterol measures I had obtained. You might have some number of them over a population threshold, 200 or whatnot. If you track the individuals over time, and I'll just draw your attention to person 25 here, you'll see that person 25 has fairly low cholesterols or legacy cholesterol values. You could establish a personal average for that particular patient and maybe establish errors. And then ask yourself, what would you do with that patient if in fact their cholesterol level deviated significantly from their legacy values, but was still below the population threshold? You could argue you wouldn't want to do anything because it was still well below the population threshold. However, if you saw a trend for increasing cholesterol levels, this might be indicative of a health status change, despite the fact it's below the population threshold. So what we're learning is that many individuals have their own baselines for a number of biomarkers, say of the type that Patrick had discussed. So this idea of using personal thresholds in the future is kind of gaining ground. A recent paper by a group from UW had studied CA-125 levels in ovarian cancer, and in this paper, which I thought should have gotten a little bit more airplay than it did, they showed that using personal thresholds resulted in the capture or identification of ovarian cancer about a year before population thresholds would have. So a pretty remarkable study. Now, how does one collect such data in a convenient way? Patrick, again, had given us a couple of things. I'm going to talk about dried blood spots quickly. A little finger prick of blood. You can now assess multiple analytes. This has been shown to be reliable, making this convenient. So someone could do this at home in the way a diabetic might do a finger prick, or the way that infants are typed for various disorders using dried blood spots. Starting to receive a lot of attention. I won't go through details, but you can use them to do, among other things, therapeutic drug monitoring studies. So we might analogously use them in therapeutic nutrition monitoring studies of the sort that Patrick, again, had alluded to. Last thing I want to talk about is this concept of vetting algorithms for matching nutritional interventions to patient profiles, which again is gaining ground. What I mean by this is the following. So let's say you have a patient, you don't know what diet to give them, right? Now there's a whole lot of emphasis on, say, nutrigenomics. So you look at their genomes and on that basis decide that they might get the Atkins diet versus the South Beach diet or something like that. Now we're moving a little bit beyond that, and instead of just using genomics, using a whole battery of omics profiling to see what kind of perturbations might be present in any one individual's profile, and on that basis decide what their diet might be. Now, the important thing here is there's got to be some kind of strategy for matching the profiles to the nutritional interventions. And so if one wants to test these, what you have to understand is what you're testing is an algorithm, not the individual interventions. And this is crucial. I'll give you an example where this is going on routinely in the cancer field, which is, again, one of the areas where I work. This is a real eye chart of a table, but just follow me here. What this does is it lists kind of perturbations often found in individual cancer patients' tumors. And what is known is that for many of those perturbations, there's a drug that can uniquely counteract the pathophysiology induced by those tumor mutations, right? So what we have are particular drugs like Herceptin. You'd give that to a woman whose breast cancer is overexpressing the HER2 gene. For individuals with a certain type of leukemia who have the Philadelphia chromosome, you'd give them Gleevec. So as a community, we're identifying all these little matches between tumor perturbations that might be unique to a small set of the population and specific drugs based on the activity of those drugs. So what the community is faced with is having to run bazillions of little trials with a small number of patients. And that's thought to be very inefficient, and no one wants to actually pay for those studies. So what the community has done is come up with an alternative strategy, one that might be applicable in the nutrition field, called bucket or basket or umbrella trials. And I'll just tell you a little bit about how those work. But essentially what you do is you enroll patients and you profile their tumor to see what perturbations are present. You identify which patients have which perturbation, and you have a number of treatment baskets. And for every patient that walks in that shows a certain profile, you then steer them towards one of the particular treatment baskets. And in this sense, what you're actually testing at the end of the day, and I won't go through all the detail here, is this algorithm for matching the tumor perturbations to the drugs. It's not the individual drugs that are being tested. This is a crucial difference between the way interventions were vetted in the past and the way they're currently being vetted, at least in the oncology field. I've talked about the implications of this with the FDA, and again, Sean Causal is mentioned up there. And when I went, we thought we would come up with a list of kind of recommendations for how we could overcome the problems associated with these trial designs. Instead, what we actually did was raise more questions than we answered. And here's some of those questions that were discussed at this briefing with the FDA. I'm just going to draw your attention to a couple here. One is, what if the trial fails? For example, there was a trial called the Shiva trial that was published in the Lancet a little while ago, where they took a bunch of individuals, did the basket kind of a trial approach, had a bunch of individuals that were guided towards treatment baskets based on their tumor profiles, and they showed no better outcomes than people that got standard of care. This was thought to be an indictment of personalized oncology. The community was in a real place of disarray, didn't quite know how to respond. Myself and other people just said, well, maybe their algorithm was faulty. Maybe they were steering people with the Philadelphia chromosome towards the Herceptin basket. So you have to look under the hood and see on what basis is your matching algorithm actually based before you indict the whole field of personalized oncology or personalized nutrition, whatever it might be. Another interesting problem had to do with insights that arose external to the trial. There are many trials like adaptive trials where you kind of rotate which interventions are given to individuals on the basis of the data that you accrue within the trial. But what happens if there's data outside the trial that might sway you? So let's say the day after you start your basket trial, there's a cover article in Nature saying people with the HER2 perturbation should get a different drug. Should you ignore that information? If you ignore that information, maybe you're violating a little bit of ethical kind of issues with your trial. So what I've done is worked with the FDA, had a trial in cancer space where we're allowed to adapt the rules over time. This proved to be sort of a nightmare for the biostatisticians analyzing the data, because it meant that the people who were enrolled in the later stages of the trial were being guided to different drugs, or guided to drugs based on a different algorithm than the people at the start. Okay? But this is a reality if, in fact, we're going to use adaptive trials or test algorithms in quite this way. Okay, now another issue that we had to debate was the nature of the algorithm itself. So you have to enroll patients, and then you have to profile them. That's through a bunch of different assays, metabolomics, genomics, et cetera, et cetera. Then you have your matching algorithm, and then maybe have a board deliberate over they really want to go with that intervention for the patient. In oncology, this is necessary. You have usually a tumor board that decides whether the drugs that were recommended by the algorithm are likely to benefit the patient. So the tumor board could override the algorithm's decision. And then, of course, you measure the patient outcomes. In the course of the conversations with the FDA, we realized that the intervention matching rules went beyond the actual algorithm, the listing of which perturbation goes with which drug, and included the assays that were used, kind of the garbage in, garbage out principle. If you can't assess the perturbations in the tumor properly, then all bets are off as to whether your algorithm is going to work. We also had to factor in the deliberations of the tumor board. So again, I could see this kind of coming to the fore in nutritional science when people try coming up with algorithms, like many companies have done. One question is, should all these algorithms that are being thrown out there by different groups actually be vetted, and vetted in a regulated environment? Huge question. Don't know that I have the answer, but it's one that's come up. Case of Watson. Watson is used often in oncology for deciding what drugs a patient should get. It's a very complicated and sophisticated system that they have. But should it be put to the test? Should one say, does Watson do better than standard of care? A reasonable question to ask. Now, to IBM's credit, they are trying to vet the utility of Watson by comparing it with different alternative ways of deciding what treatment someone should get. There are many companies in the nutrition field that are proposing algorithms. So Pathway Genomics in my backyard in San Diego, Habit, Imaging Labs, all have services where you pay some money, you have your genetics profiled, and then they'll give you recommendations on what diet might be optimal for you. They all have slightly different algorithms. Should there be a scientific bake-off between these algorithms? This is a reasonable question to ask. So in answering this question, should there be regulated vetting of these algorithms? I would argue maybe not, but any group or company that is so incurious about their algorithms, not to want to show that it works, you might want to be skeptical about. Last thing I'll talk about in kind of the context of personalized medicine is there is this belief that for people with at least rare conditions, the best way to approach treating them might be to convene a panel of experts with different areas of expertise at the table. So you might have genomic scientists come in, right, to understand what perturbations they might have. Maybe in cancer, this is a little bit of a bigger issue. Then have to decide what those perturbations do and what interventions might be the most appropriate. You have to then decide on an intervention. You then have to decide on a trial to convince yourself, an N of 1 trial, that whatever strategy you came up with is going to benefit the patient. And of course, then you want to record the data. I would argue that some of this stuff we have down. Other stuff, it's iffy. Deciding what the pathophysiology behind an individual patient's disease might be is complicated, but there are emerging technologies. We do need to have a session on, say, organoids or iPSCs with CRISPR and stuff, but they're very relevant to determining what pathophysiologies a patient might have. So I'll just stop there because I'm going over and conclude that N of 1 trials are here to stay. Personal thresholds will be used. We may have to vet algorithms in the future, and I think it does make sense to convene teams, especially for people with rare disease, to figure out how we can deal with them. And I'll just acknowledge my funding. Sorry, I went over a little bit.